glycolysis or glycogen metabolism? Good question. So always think about phosphorylation as a switch. So as we've got there, phosphorylation acts as a reversible switch, and that's one of the benefits of phosphorylation, is that you can add it, you can remove it, and depending on which state you're in, you're either in the on state or the off state. And typically, and this is not always true, as we'll see, that when you add a phosphate, you turn on an enzyme, and when you remove the phosphate, you turn it off. That is not always the case, but that's by and large, if in doubt, that's usually what is occurring. So acts as that switch because the kinetics of it allow you to modify the presence of the phosphate very, very quickly. So it can persist or it can be there for a very short period of time, really depending on the needs of the cell and on the pathway. So reversible switch either activates or inhibits enzymes in response to cellular signals. So remember, the cell is going to be responsive to a particular pathway. And this is one of the other questions that we've got coming up too, in terms of how does the cell know where things come about? And really it's about substrate needs and responding to the environment. We know that regulation is crucial for coordinating metabolism, because remember, metabolism is not a fixed thing. So your energy demands will change. If you're resting, your energy demands are quite different than if you're running a marathon. And so that has to be taken into account, and the metabolic profile also has to respond accordingly. You know that you've got hormones such as insulin, glucagon, adrenaline, they all change the response. And again, these will vary depending on the environment. Hopefully, levels of adrenaline are quite low when you're resting, but when you're in an activated state, or perhaps in a different type of condition, adrenaline levels might rise, and therefore you need to respond accordingly. And of course, there's nutrient availability that's going to affect that. So, two examples. So glycolysis and glycogen metabolism. So glycolysis, you've got PFK. Now I've gone into more detail here, but I think it's just really important just to help understand and explain the concept. So you've got PFK2 and fructose 2,6-bisphosphatase. So phosphatase is telling you that it's removing it, the kinase is adding it. So remember, you have the forward reaction, you have the reverse reaction. These control the level of fructose 2,6-bisphosphate, and you know that that is a potent activator of PFK, which is a key enzyme in glycolysis. This also touches on a lot of the questions that I've got from students, and by a lot I think just the ones that are here, the level of detail that you require. Remember, with metabolic pathways, the rate limiting enzyme, the control points, the points of regulation are always the critical aspects. The focus is not always as detailed into the entire pathway. Okay, it's more about what is it doing, why is that pathway important, how is it controlled, etc., as opposed to the individual specific details. So in this context, phosphorylation is stimulated when you've got glucagon. That inhibits PFK. And so this leads to a decrease in glycolysis. And obviously, if you reverse the effect by taking away the phosphate, then you increase glycolysis. So there again, you've got those two control mechanisms. And similarly with phosphorylation of pyruvate kinase, which is that last step in glycolysis, and again, this is a critical enzyme. In the liver, it's stimulated by glucagon. And this inhibits pyruvate kinase activity, and this stops glucose being used during fasting. And again, dephosphorylation by insulin restores activity. So 2 points here. One we've already covered, which is that the kinase is going to do one thing, you have the corresponding phosphatase to do the opposite, but also notice the involvement of glucagon and insulin, two different hormones that regulate the two different pathways, or the two different responses that we see. Now, glycogen metabolism. So glycogen phosphorylase is what breaks down glycogen. And so you need this when you have an energy requirement. So this is going to be activated by phosphorylation in response to glucagon or adrenaline. But again, we've got now both doing the same thing, but notice the different location. So adrenaline in the muscle is more associated with an energy expenditure, activity, exercise, running away from a tiger, those sorts of things where you need to move. And this mobilizes glucose during that exercise or the fasting period. Glycogen synthase generates glycogen, so the opposing side, inhibited by phosphorylation. So here's an example where phosphorylation is not doing the standard approach, it's doing the opposite approach, but it's activated when it is dephosphorylated, and this is promoted by insulin. So again, those three key molecules, glucagon, adrenaline, insulin, are here, and you can think about phosphorylation. Well, why does it activate or why can it inhibit? Remember that it is giving you a molecule with a high net negative charge. That negative charge can lead to a conformational change in the protein, and that conformational change can either activate or inhibit the enzyme. And that's really the key point in terms of that idea. Now, I'll quickly just go back to, I'm sort of flipping between here and the presenting mode. Can I just check with you all? Does that make sense? Is there anything that is not clear before I move on? As I mentioned, I'm sort of time constrained. Thanks, Tamara, Abby, Ira. Okay, excellent. Thanks for the feedback, everyone. Okay, sorry, I'm only working one screen, so I'm going backwards and forwards. Okay, so next one. What determines the specificity of a kinase for its target protein or amino acid residue? Now, before I answer this, also just to give you an idea, I've answered these questions as they've come through. So these questions might jump from one topic to another, but that's okay. It's really just talking about these were the areas of concern or questions that individual students had. So kinases, as you know, recognize short conserved sequences, which are called a motif. And that sequence, if it is restricted to a particular substrate, it limits the activity of the kinase. Similarly, if that motif is shared, that kinase has multiple substrates that it's able to act on. In terms of the motif, you typically see serine, threonine, or tyrosine residues as where the phosphorylation is added. And when we look at it from a signal transduction perspective, particularly with tyrosine kinases, it's in the name. You know what the substrate amino acid is going to be. It's going to be tyrosine because it is a tyrosine kinase. In terms of other aspects, you've got the 3D conformation of the substrate and the kinase, because remember the two have to come together. And this determines whether the substrate can actually fit into the kinase, into the active site. So if the kinase is turned off, its active site is going to have a different configuration and the substrate may not fit in. On activation of the kinase, as we've seen with receptor tyrosine kinases where they can cross-phosphorylate each other, that leads to a conformational change, and that in turn is going to allow for the substrate to gain entry into the active site. So that's one control. Kinases are also going to be regulated by actually where they are within the cell and also where the substrate is within the cell. So if you've got a kinase, like a receptor tyrosine kinase, which is embedded in the cell membrane, you need your substrate to also be in the cytosol, right? So think of it this way. If your substrate for a receptor tyrosine kinase was in an organelle, is it going to be accessible to the receptor tyrosine kinase? It's almost as if it's encased, isn't it? So you have to have the substrate in the same location. So in those contexts, it needs to be in the cytosol. Now, if you have a receptor which is within an organelle, then you can still have that activation of the receptor at the organelle level, but again, within the organelle, you're going to need all of those downstream molecules. And lastly, some kinases are only active when they're bound to cofactors. Now, in this context, this is probably not the best example, but we know that some protein kinases, so remember the C stands for calcium, require calcium ions and lipids like phosphatidylserine in order to be activated. Now, there are lots of different isozymes of PKC, and not all of them need these two requirements, but the most common ones certainly do. So again, that really determines the specificity and activity of a particular kinase. And of course, feel free to jump in, raise your hand, unmute yourselves, do whatever you like to interrupt. And I don't mean interrupt in a bad way, but to stop me if there's a question or anything else as we're going through. So again, I'll just check with you all. Is there any comments or anything? Was that clear? Thank you. Thanks, Charlotte, Tamara. Okay, so let's move on to the next one. Explain the steps involved in the initiation of protein synthesis in eukaryotes. How are they regulated? Now, I have given you an answer here for the student who asked this question, but one thing that I would preface is you should know what's happening with prokaryotes first, because prokaryotes are undergoing the same basic process, but eukaryotes are just more complicated in terms of the idea. So what you'll see here is that the ribosomal subunits are bigger, so 40S. They also bind initiation factors, but there's probably more of them. You start off with an initiated tRNA, which again is going to be methionine, but remember it's not formylmethionine. That's one of the key differentiators between prokaryotes and eukaryotes. They're both loaded with GTP because you need energy. But again, notice here that it is GTP as the energy source, not ATP, which we've seen typically. We don't have a Shine-Dalgarno sequence with eukaryotic cells, so it's typically the very first AUG, which is for methionine, that comes into play. And there was a question, I think, later on that does talk about what level of detail do we need to know about amino acids and in this context. Certainly do not expect you to know the triplet codons. The only reason I know the AUG codon is because it is for formylmethionine. We talk about it often. Okay, so don't go on and think, well, I need to know what the triplet codon is for serine, for example. There's a table for that. You can look it up. You don't need to memorize it. The binding then leads to the hydrolysis of GTP, then brings in the larger subunit of the ribosome, and then you form again the larger ribosome complex. So again, really very similar, just bigger ribosomes. And it's now at the start of the message, or where that first AUG is. It's in the P site because you've already got that first amino acid, because remember the P site is for the protein, and then the elongation phase continues. Now, the other point was about regulation. I don't go into regulation. It is there. There are initiation factors. There are elongation factors that can control protein synthesis. That is much more detailed than you need to know. It's probably a third year level, if not higher level sort of detail. So I've not given that to you on purpose. We don't need to go into that sort of content. Okay, again, back to you all. Let me know. Is that good, bad, any points of clarification? Thanks, Tamara. Ira, thank you. Now, Ira, is that a hands up or thumbs up? Okay, perfect, thank you. Just good to clarify. Okay, thanks everyone. I appreciate you responding so quickly. Okay, how do post-transcriptional modifications of mRNA, like the 5' cap and poly-A tail, influence the efficiency of protein synthesis? So this is a really good question. You have got these two different types of modifications. So the 5' cap, which is methylguanosine, this protects it from degradation, and so it actually facilitates binding of the ribosome. So again, most of these features are going to have multiple characteristics, so it's not just one thing. Facilitates binding. As a result, it also promotes efficient translation and initiation, so the whole starting process. If you cannot bind to the ribosome at the beginning, you're not going to be able to read the message, and that whole process is not going to occur. And then similarly, it enhances that recognition of the start codon to the AUG. And lastly, it protects the mRNA from degradation. So really the last one is the one that we focused on mostly, but given a bit more context and a bit more detail here as well. In terms of the poly-A tail, you remember this is not encoded in the genome. You don't have a string of A's in your nucleic acid. There is a poly-A enzyme that will add on the poly-A to the mRNA. This enhances efficiency, and it certainly stabilizes and protects the 3' end from degradation. So you can think of the methylguanosine as protecting the 5', the poly-A affecting or protecting the 3', and it also, and I haven't written it, which is an omission, it also extends or can affect the half-life of the mRNA. So depending on the length of the poly-A tail, the messenger RNA may exist for longer or shorter periods within the cell. That's important because if you've got a message which is short-lived, it may be that the cell only requires a small amount of protein or product. if you've got a cell that requires an extensive amount of product to be generated, then you want the half-life of that message to be longer, so that you're not constantly always going back to the DNA, having to unwind it, rewind it, etc. Okay? So again, back to you all. Any questions, or is that okay? Thank you, thank you. Okay, perfect. Okay, I won't go through this table. This is a much more detailed table, but again, differences between prokaryotes and eukaryotes. So just very quickly, we've covered much of this. Cellular location, so looking at cytoplasm versus organelles. This is really looking at the difference between prokaryotes and eukaryotes. Structure, so there's no modifications for prokaryotes, only eukaryotes. Half-life, short versus long-lived. Processing, so this is about splicing. So you can only splice when there are introns. There's no introns in prokaryotes, so that doesn't occur. Transcription translation, so it occurs at the same time. This can't occur within eukaryotes because you're in two different locations and you've got organelles. Formylmethionine versus methionine. The size of the ribosomes is slightly different, so it's slightly bigger. The importance of the Schindel organo versus the first AUG. More factors versus less factors in eukaryotes versus prokaryotes. And prokaryotes, you can have one message with lots of proteins. You don't see this typically with eukaryotes. You have one protein comes from one mRNA. And a lot of these characteristics that we see with prokaryotes also go on to explain why they can replicate so quickly, because they have less constraints than other cells or eukaryotes. Okay, so this question was about DNA supercoiling and this image in particular. So is it depicting supercoiling? So not quite. This diagram is showing you the initial stages of DNA unwinding, and showing you that first bit, which is where it is unwound. If we open up that DNA at that location, we're adding stress to the other side. And what is really important is that, and I'm going to jump to the fourth point down here, this really relates to bacterial genomes in particular, because they're circular. And what you need to think about, and one of your bits of homework, is get a rubber band, pin one end down, okay, and hold it, and then twist the other end. And because it's circular, you can imagine it's two chains coming together. As you start to twist it, you're going to introduce stress. Eventually you'll get to a point where there is so much stress in the RNA, it's going to collapse on itself. And that collapsing is really the formation of those supercoils, okay? It's dealing with that stress. So you have regions that are unwound where there's less stress, and this other region on the right-hand side which has more stress. So remember, helicase, so the important point is what enzymes do this. So helicase unwinds, topoisomerase leads to the release by cutting and unwinding. And one way that a cell can deal with this is through supercoiling. When you do molecular biology and you're looking at nucleic acid, you may see nucleic acid, particularly with bacterial genomes, that are supercoiled. Because they are smaller than the unwound genome, they're actually going to migrate a lot quicker on a polyacrylamide or an agarose gel more likely. And so they'll seem to be smaller. It's not that they're smaller, it's just that they're more tightly wound, so they're taking up less space and therefore migrating more rapidly down the gel. Okay, so back to you all. Does that make sense? Has it clarified the comment? Okay, excellent. Thanks, Jordan. Thanks, Charlotte. Okay. Now this is a common question. And I understand where these questions are coming from. What precursors, molecules, enzymes do we need to remember for the nucleotide biosynthesis for pyrimidines and purines? Bicarbonate, phosphate, etc., blah, blah, blah. I know that this lecture is a very complex lecture. There's a lot of players, there's a lot of things that are going on. And this comment will be true not only for this lecture, it's true for all the other lectures, and it will be true for all of the future lectures if you're doing immunology with me in SP5. Focus on the big picture first, okay? What is the point of the nucleotide biosynthesis lecture? The point is that you can either make nucleotides or you can recycle them. if you want to, why would you go down either pathway? Well, if you make them, it's because the organism has a requirement, so their need is greater than what is coming in through a diet. You can recycle them because you've got cells that are dying, or it is coming through a dietary process. And just as we would say with, say, an aluminium can, it's easier to recycle the can than it is to go and mine the aluminium and process it to then make a new can. But we do it because there is a demand. The next point is that simple precursors like bicarbonate, like amino acids, like ATP are used to make complex molecules, right? You look at the nucleotides and they look quite complicated, 5 rings, 6 rings, double rings, etc. What you need to appreciate is the carbon, the nitrogen can come from very simple molecules like CO2, like amino acids, like ATP can certainly drive the process. You then also need to remember that all of the bases that we make, and here I'm talking about the diagram, the 2 summary diagrams, and I've got that on the last point, they all finish on an intermediate, a common core, which is a precursor that leads to the 2 RNA bases. So every cell will make an RNA base first, and then to convert from RNA to DNA is a simple reaction because it's only the difference between ribonucleic versus deoxyribonucleic acid. When we go through the processes, we've seen that phosphorylation can be one way of mediating a reaction. So you phosphorylate and then you have an amine group that comes in. The point of adding the phosphorylation is a target for reactivity. When you look at these molecules, there may be lots of different points where a point of reactivity can occur. How does the cell, from a chemistry perspective, how does the cell ensure that a reaction occurs on, let's say, carbon number 3 as opposed to carbon number 1 or 6? It does so by putting a group that is more reactive. And in this case, it's going to be the phosphate group. So that is going to be the point of control. And when we saw that, we saw phosphorylation followed by amination and deamination as a point for reactivity and conversion. So that's something of commonality. You need to think about the importance of the salvage pathway. One thing that I haven't got here is you also need to appreciate the point of interfering with nucleotide biosynthesis, the different drugs, the different mediators that can be used and why that would be used. And so here we're talking about cancer, where you've got uncontrolled cell growth. One way of interfering with that is through different inhibitors. And again, thinking forward, when you come to do immunology, we will talk about that in the same context, but we'll use it where we're trying to make antibodies and we actually want to interfere with that antibody process. And again, go to those 2 summary diagrams of the purine and pyrimidine pathways, not because you need to memorize the steps, but because they outline all of those key points. Okay, they highlight all of those key aspects and key ideas. Okay, really want to hear now from you all. Does that make sense? Do you now understand again the focus for this lecture and not sort of getting stuck in the detail? Thanks, Ira, June, Charlotte, Tamara, thank you. Okay, Callum and Abby, are you guys okay? Thanks, Abby. Callum, are you good? Or do you have any points of clarity you want me to cover? No? Okay, no worries. Okay. So again, with all lectures, with all courses that you do, sorry, it's in the chat. All good, okay, thank you, thanks Callum. With all content that you get across all your courses, always focus on the big picture first. Go back and think, what is the whole point of this lecture? Right? What's the point of this lecture? And then what are the level of detail? If you don't need to have all of the detail, don't go into that level of content. And you might be thinking, well, why do you being me, the academic, why do we give you this detail? It's part of the story, it's part of explaining the process, okay? We can't just give you all the bits that you need to know because otherwise you're not going to understand how it works in context and how it all relates to each other. And the really good example that I like to give, it's like you're reading a murder mystery book. If the book had one page that said there was a husband and wife and a butler, the husband had a lot of money, the butler killed the husband, and he was then found guilty. That's not really much of a story, right? That's where all the other information comes in, to give you a bit of context and to explain why did the butler kill the husband? Why was the wife unhappy with the husband? I'm sort of going off on a tangent here, but you can see, hopefully, that you understand the context, that you've got to have this extra information to really understand how it all fits together. And that's what this lecture in particular is talking about. Certainly don't want you to go through the individual steps or remember the third reaction that was involved in the making of purine or pyrimidine. No. But the hyperxanthine guanine phosphorybosol transferase, as an example, is a critical molecule. That's one that you should know. Why? Because it has such a central role. And again, I would have highlighted that to you in the lecture notes. And certainly in the lecture recording, I did highlight that to you. So there's lots of points of direction that we give you to make sure you understand where the important parts are. Okay, so move on. Now the next one was about cholesterol synthesis, and it was just more of a generic question, I think, because the student that asked this, I don't believe, is here. So the key points, remember the key points of cholesterol is that it's essential. We make it, so if we make it, we must need it in our bodies. But most of the time, much of it comes from our diet. It is important because it's a precursor of all steroids. It's a component of the cell membrane, and the cell membrane affects the fluidity of the membrane. And it can even contribute to bile formation, which is to do with, obviously, with digestion. So you can see quite diverse activities. You do need to understand how it's transported throughout the body, because this is an important feature. Everyone knows that if someone has high cholesterol, right, that's never a good thing, because it's always the bad cholesterol that is elevated, and we want to change it to the high density cholesterol pathway. In terms of synthesis, and again, this really relates to the previous lecture, what do we need to know from the synthetic perspective? First point, all the carbon atoms excuse me, are coming from acetyl-CoA. We have used acetyl-CoA to make cholesterol. We've used it to make ketone bodies. We've used it as an output from beta oxidation or lipid metabolism. It's part of the citric acid cycle. Okay, it's one of those key molecules. So it's the same process as making ketone bodies, it's just in a different location. So this is occurring within the cytosol. So you've got mevanolate as that first committed step, and it's occurring in the cytosol. So the two key points here is location. So cytosol versus mitochondria for the ketone body, which is on the next line. And then why have I shown you mevanolate? It is the first committed step. Do I really place a lot of emphasis on all of the other enzymes in between? No. Why? Because mevanolate is the most important one, really. If you want to control the pathway, that's where you target. The enzyme that is involved in that first step is the point of target of cholesterol-lowering drugs. Okay, so again, you can see why the focus is on these particular molecules as opposed to the second or the third molecule. So the HMG-CoA reductase, which is that point of control, is important because it controls that first step. It is part of that rate-limiting enzyme. So again, use this same logic in all of my lectures, and probably in other lectures as well, other courses, but this is really the key points of focus. Okay, back to you all. Is that okay? Okay, excellent, thank you. Good, thank you. Okay, next one. Beta oxidation and ATP generation. And I'm pretty sure the student that asked this is online, which is great. Now, this is one area that is not a difficult concept, but students will make mistakes very easily because they forget key points. So I'll go through it, and there's an example calculation on the next slide. When I finish, as always, if you've got any questions, please do highlight. So beta oxidation and ATP. Remember, we only talked about even fatty acids, and we only spoke about saturated fatty acids. So you can immediately ignore unsaturated fatty acids and also odd carbon fatty acids. Do they exist? Do they get metabolized? Yes. Is it beta oxidation? No. It's actually omega oxidation. It's a different pathway. We're not going to worry about that. We're just going into the saturated pathway. Basically, you take your saturated fatty acid and you convert it into activated acetyl-CoA. Activation, we've always spoken about, is merely adding a phosphate group, okay, making it more reactive. Those molecules then enter the citric acid cycle, and you know that the citric acid cycle ultimately gives rise to ATP. There's multiple steps. There's oxidative phosphorylation, there's the proton gradient, but ultimately you generate ATP. Now, I've given you 4 metabolic steps, but remember, there are 2 previous steps that have to occur. The first step is the activation of the fatty acid, and then the second step is the transport into the mitochondria. They are 2 essential steps. They are not involved in the metabolism per se, but without them, the fatty acid will not get into the organelle where oxidation takes place. Okay? So, what's an example? You might mix flour and milk and eggs and, actually not milk, but or you could, to make some bread. But if you don't put that bread into the oven, it's never going to cook. Okay? And without those 2 precursors, you're never going to get bread because that's part of the process. This is the same idea. You need those 2 steps to get it into the right location. Once it's in, what do you have? You've got oxidation, and this introduces a double bond. Why would you want to introduce a double bond? Because that makes it more reactive. Remember, oxidation is loss, so FAD comes into play because it takes it up. You've got a double bond, it's more reactive. What do? We hydrate it with oxygen as the point of reactivity. It then becomes oxidized again. Oxygen that is oxidized goes to an alcohol, well, from an alcohol to a ketone, and now we have NAD+. Those 2 molecules are going to be important because they're part of the calculation. And then right at the very end, we chop the fatty acid using coenzyme A, and we yield an acetyl-CoA unit. Acetyl-CoA is 2 carbon atoms, so the fatty acid is now 2 carbon atoms shorter. Notice it's 2, and notice that we're talking about even fatty acids. So you can go from 20 to 18 to 16 and so on. If you had an odd, you might go from 15 to 13. Eventually you're going to get to a 3 carbon unit, and that's going to be problematic. So don't worry about that for now. So the next idea is, what do you generate per cycle of beta oxidation? So as we said, acetyl-CoA is shortened by 2 carbon atoms, so the acetyl-CoA comes off. You've got 1 molecule of FADH2, which we had in step 1, a molecule of NADH, which we had in step 3, and the acetyl-CoA, as I mentioned, each one of those is formed. Each one of those, and I've written worth in sort of quotation marks, each one of those contributes a certain amount of ATP. This is what you need to remember. FADH2 is 1.5 ATP, NADH has greater potential, so it's worth 2.5 ATP molecules. And acetyl-CoA is 10 ATP molecules. This is the first part of the process. You need to know, okay, what does that equate to and how much ATP? The next step is then, well, how do I work out how much ATP is going to be made? I could pick any number, and I've given you 18 fatty acid here. So this is, it's not oleic acid, but it's the saturated version of oleic acid. So what you need to do is, first of all, determine the number of cycles. So it's the number of carbon atoms, 18, divided by 2, because we chop it up 2 at a time, minus 1. This is where it's going to perhaps sometimes confuse you. Why is it minus 1? If you imagine we only had a full carbon fatty acid, so butyrate, if we chopped butyrate into 2 carbon atoms, you generate 2 acetyl-CoA in that last step. So you don't need to take a 2-carbon, go through beta oxidation again just to generate a 2-carbon cycle. So we have to subtract that very last process because we get a bonus acetyl-CoA coming out. So 18 divided by 2 would be 9 cycles, minus 1 would be 8 total cycles. Now, we do still need to remember that that last cycle gave us a bonus acetyl-CoA, and we must account for that. So when we look at the equation, or the calculation, 8 cycles times 1.5 is 12 ATP. 8 times 2.5 from the NADH is 20 ATP, but it is 9 acetyl-CoA, because remember 8 cycles, but that bonus one, so 9 times 10 gives us 90 ATP, and that's what I've written here. You add them up, and your initial subtotal is 122, but remember, the first 2 steps was activation and transport. Activation was ATP use. So you have spent some ATP. So therefore, the initial energy or the total amount that you generate is actually slightly less. So you have to subtract those 2 from the total, and your absolute amount is 120 ATP molecules that is generated. Students will make 2 mistakes. They will forget to subtract the ATP that has been added, or they will forget to account for the extra acetyl-CoA that was generated in that very last step. Okay, I'm just going back to the screen. Let me know, does that make sense now? Okay, excellent. Is there any point of confusion with this calculation? Anything that you'd like covered, or it's all good? Okay, perfect. Thank you. It's a great question. It is a common area of confusion. Okay. Now, I think the student that asked this question is also online, which is great. Concept of allosteric control and inhibition, how is that just different from inhibition? So good question. So allosteric control refers to the regulation of an enzyme by binding of an effector molecule. So it may not be the substrate. And that effector molecule can either activate or it can inhibit, but it does so at a site not at the active site. It's at an allosteric site. So what you need to appreciate here is that when you have an enzyme, you've got an active site. Active site is where the substrate comes in. If we're talking about a simple inhibition, then if we compete for binding to the active site, yes, we will inhibit the enzyme. But if we have another molecule, let's say, I don't know, a drug, that binds to the opposite side of the active site, and in doing so it changes the shape, that change of shape leads to a conformational change of the active site. And in turn, it regulates the activity. So yes, we will affect the activity, but it's via 2 different mechanisms. Okay, it's not the same idea. So that's what that second point is talking about. Conformational change can either increase, so activate, or decrease or inhibit the function. And when I was thinking about what we've covered, we can think about Gleevec, where we were talking about the cancer lecture. Inhibition can refer to a drug like Gleevec that competes with the normal substrate. This is an example of competitive inhibition since it affects the active site, but this is not allosteric control. We've seen allosteric control where we've got the product of an assay or of a reaction coming back and binding to the enzyme and preventing the substrate from coming in. That's an example of allosteric control. Similar, but different points of focus. Now, does that make sense? Okay, thanks, Hannah. Tamara, Callum, Ara. Okay, excellent, good. Okay, for DNA, RNA, protein synthesis, do we need to remember the characteristics of the promoter sites? Start codons, termination signals, or stop codons? And then the second question, which is very similar, what specifically do we need to remember about the Pribnow box, TG, TTG, ACA, Carter box, cat box, their name, sequence, and location? Okay. So, as I've got there, a high-level understanding is required. So their role, what is a codon? What is a transcription factor? What is a promoter? What are they doing? The basic structures like a tartar and a cat box, it's in the name, so it's very easy to remember. Tartar is T-A-T-A, a cat box is C-A-A-T. I probably won't ask you to have that level of detail, but it could be in a multiple choice question, for example, where I tell you what it is, but I just want to know, what does it do? The Shine-Dalgarno sequence, for example, why is that important? It's important because is it in prokaryotes? Is it in eukaryotes? Well, it's only prokaryotes. What does it do? It's involved in positioning the messenger RNA within the ribosome to make sure that the right AUG is used. You don't need to remember the codons. As I mentioned before, AUG we remember because we talk about it in terms of methionine or formylmethionine, but you certainly don't need to remember any of the other sort of sequences, etc. So again, this is another idea of make sure you understand the big picture. What are the things that we're trying to do? What do these molecules do? Remember, these are all about positioning and telling the cell, where is the gene, where is the start site, and how can I copy it? Right? Where do the proteins that facilitate transcription, where do they need to bind? That's what these things are doing. We've given you a bit more detail to say, well, what are they? Well, these are common sequences. These are commonly found regions within a gene. And so if you know that a tartar box and a cat box are found with each other and that they're approximately 15 to 20 base pairs upstream of the start site, you can then go search for a gene. You can look for that sequence, like within a Word document. We go Control F to find. Let me find this sequence. Once I can find it, I then know, well, here's a gene, potentially. So really, that was the idea that we're trying to cover. Okay, so back to you. Does that now make sense? Okay, excellent, thank you, perfect. Lacoperon, everyone's favourite. I think you would have had lacoperon in first year as well. So lacoperon study aims. How is it turned on by lactose and off by glucose? Okay, let's actually start off with the off state. It's probably easier. So I've done it at the bottom. Remember, we talk about the lacoperon as an example of an inducible system, which means that we can induce the expression of genes in this context. So there's at least three. We can induce the expression of these genes by an external environmental factor. And in that case, it's going to be lactose. So in the off state, the lacoperon is turned off when lactose is low or glucose is high. So why would we want to turn it off when lactose is low? The whole point of the lacoperon is to generate enzymes that metabolize lactose into glucose. Remember, glucose is the preferred carbon source for bacteria. They can use others, but they don't want to if they don't have to. So in this context, what it's doing is it's taking lactose sugar, converting it into glucose. If you've got lots of glucose, do you need to go and look for lactose to convert it? No, right? It's already there. If you've got, let's imagine you're hungry, okay? And I give you some flour and some water and some yeast, or I give you a loaf of bread. Are you typically going to go and make the bread? Or are you just going to eat the one that's there, the more readily option, right? You're going to do that unless you're really into baking. But if you're super starving, you're not going to wait for several hours. You're just going to go straight into eating the bread because it's there. That's the same idea with the glucose. The bacteria is just going to use the glucose. There is no need to make the lactose. It is also very energy intensive for the cell to do it. The way we turn it off, or that the cell turns it off, is that it has an inhibitory protein, which is called the I protein. It binds to the promoter. So think to the previous question about the tartar box, cat box. It binds to that region and it physically blocks transcription. So if the RNA polymerase came along, it has a roadblock in front of it. It has a gate and it's not able to move forward. So we turn it off. So yes, there is this turnover of the I protein, but the I protein can be long-lived. because it doesn't really have to do much, it just has to sit on the DNA. It can be removed when the lac operon is required, and this is then the on state. So the on state is really the opposite of the off state. The on state is where you've got a lot of lactose or you've got very little glucose. And so now there is a requirement for the cell, the bacterial cell, to get a carbon source. So lactose is the inducer. It turns on the protein or the whole process. When it is present, it goes into the cell, it is able to bind to the inhibitory protein, and it causes a conformational change in the I protein. And because of that conformational change, its affinity and its binding to DNA is reduced. So it can no longer bind to the promoter. And as a result, RNA polymerase is able to bind, and you've got the whole process moving forward. Don't also forget that cells have signal transduction pathways, and cyclic AMP is one of those transcriptional signal transduction molecules that is involved in signaling to the cell. And so when cyclic AMP goes up, it binds to a binding protein. That binding protein facilitates the binding of more RNA polymerase, and you get that 50-fold increase in transcription. Why does it do that? It does that because if the bacteria is in a stressed environment where there's not a lot of glucose and only lactose, it wants to convert that lactose to glucose as quickly as possible. So it needs to turn on the lac operon as quickly as possible. And so having more binding of the polymerase facilitates that process. And it does so because it does not want to limit its growth, its energy production, and its source of access to carbon. And that's really, really important. Okay, back to you all. Does that now make sense? Okay, thanks, Tamara, Ira, Hannah, Callum, June. Okay, perfect. Okay, we're nearly done. Okay, relating to lecture 3. With binding affinity, you talk about that at higher levels of glucose, the protein preferentially binds to the glucose in the buffer instead of the column, the glucose attached to the bead. Correct. Don't understand how the proteins will change what glucose they're attached to. Is the buffer glucose and the bead glucose different? Okay, that's a really good question. So the answer is no, it's not different. It is the same molecule. So this is referring to affinity chromatography, where you've got a molecule that has high affinity to a substrate, and the example here is just glucose. So what we saw was that as the concentration of glucose rises, there's a point where the protein, and I've put it in question marks or quotes, prefers to bind to the glucose in solution. There is more, the affinity is still the same, but there's just more of it, so it's easier for it to bind to what is there. What you have to appreciate though is you might think, well, hang on, if I'm attached to something, I'm attached. And that's correct, but what you also need to appreciate is that proteins don't just bind and stay bound. Protein ligand interaction, like glucose, is not permanent. Proteins will bind and unbind to the ligand continuously. And this is based on equilibrium, so the amount of the glucose that's in the column, and also the affinity. So the affinity leads to its release from the column, and in turn the binding to the glucose, because the equilibrium is now shifted to what is in the mobile phase, higher concentration, so it's now shifted towards that environment as opposed to what is a fixed concentration on the beads. It's really the same concept as we saw with the sodium chloride. Here it was more, we disassociate, and so there's more out-competing. But again, if it's bound, how do we remove it? Well, if it's binding and unbinding, eventually you're going to have this unbound state binding to an equilibrium and also a concentration of either the positive or the negative ion in the mobile phase. And therefore it's going to be removed off the column and dislodged. Okay, does that make sense? Okay, excellent. Thanks, Charlotte. Tamara, Ira, June, Callum, Abby. Excellent, thank you. It's a really good question, and again, it's one of those things that we don't go into a lot of detail. It's easy to think, oh, it binds and it's attached. But what's happening is that it's binding, unbinding, and it's this constant flux. Okay, last one, I think. Lecture 20, tutorial question 1. Don't think primitive biosynthesis regulation was talked about in the lecture, but I wrote down what you talked about in the tutorial: substrate mediated inhibition of the enzyme, and that's an example of allosteric control. Excessive products, there's no need for synthesis and homeostasis is maintained. Correct. That's core to all of the enzyme processes that we talk about. So with allosteric control, the product can come back and inhibit. You can have, if you've got excess product, it comes back and that prevents excess synthesis, and it also helps to maintain a homeostatic level. So if you imagine that the cell requires 1 millimolar of a product, if it's at 0.1 millimolar, it needs to produce it, so it starts to go up. As it reaches that point, the enzyme will start to turn itself off, but typically it will overshoot. So it might go up to 1.2 millimolar, and then it will stop, it'll get consumed, and then it will go back down. So it's a bit like a sine wave, goes up and goes down. The tighter we can keep the concentration around that preferred concentration, the better the homeostatic control there is for that molecule and for that pathway. And that's really what the cell is always trying to do, is trying to keep it as tight as possible. So, in terms of my answer, regulation mostly occurs at the beginning of a pathway, and we saw that with cholesterol, where we had the CoA enzyme, the reductase enzyme, that was involved in that conversion of vanillate. But it's also coordinated with energy levels and nucleotide demand in this context. So if energy is low, you're not going to want to go down this pathway. And similarly, if you've got a lot of nucleotides, then you're not going to synthesize a lot of molecules. But in terms of this process, carbamyl phosphate synthetase is that first committed step in de novo pyrimidine synthesis. This is where you've got simple molecules. Carbon dioxide, ATP is the energy source, and glutamine, an amino acid. And this gives you carbamyl phosphate, and that's that first step towards making UAP. As those products increase, as we mentioned, this allosteric control that turns off and decreases the pathway. So we've got a feedback loop, and in this case, it's going to be a negative feedback loop because we're turning things off and inhibiting that process. Sorry, I've got to bring the noise outside. Okay, last point. So back to you now. Does that now make sense and help clarify the same sort of idea of that background control? Thanks, Jim. Tamara, Abby. Thanks, Charlotte. That's good to know. Okay, so that brings me to the end of the tutorial. I don't know if I've got anything else. It's only 17. Is there anything else that you would like to ask? Either feel free to unmute yourselves. It's only 7 of you, or in the chat function. You're all good? Give me a thumbs up if you're all good. Okay, good. These are really good questions, by the way. And a lot of the questions that I got were very detailed, showing a lot of thought, a lot of engagement with the content. And I think it's really important to highlight that and talk about that. How about ATCase? Hannah, what do you mean? Is it regulatory too? There will be, most enzymes will have a regulatory point. So there will be a point where you can regulate activity further, moving further and back. That was just one example that I've used in this pathway. Yeah, but that's a good question as well. All right. If there's no further questions, I'll give 5 minutes back to your day. I've got a meeting at 11 as well. Good luck with the exam. Remember, you can keep asking questions up to the exam, probably Saturday, maybe try Sunday. That's probably at the latest. Probably won't reply on Monday. It's just going to be way too late to do that. Chat, thank you. Oh, excellent. Thank you. You're all welcome. Have a great day, and I'll see you all on Monday. Okay, bye now.